Hello, my name is Taylor McGee. I'm a rising sophomore at Hampton-Sydney College, and this summer, working with Dr. Hargadon, I investigated how variable levels of mRNA, or protein-coding RNA, influences a cancer patient's survival. One of the important things I have to address before we get into some of the nuts and bolts of my research is this essential question, what is cancer? And so within the context of this presentation, cancer is an umbrella term that refers to any group of abnormally dividing cells. And within this context, it's incredibly important to acknowledge the fact that every individual tumor is existentially different. And one of the ways that those tumors can be existentially different is by having variable levels of protein coding, or mRNA. The Cancer Genome Atlas is a wonderful tool that's a free resource available online that is a revolutionary data collection program that was started in 2006. Since their beginning, they've collected about 2.5 petabytes of data, each of which give researchers a unique profile of over 10,000 different patients. And so one of the things that makes the Cancer Genome Atlas very unique is that you have the ability to partition the database as you need it based on certain cohort factors, but you also have many programs like the KMplot tool that I used, developed by Dr. Balazs Gyorfi, to make really powerful downstream interpretations without actually having to do any of the manipulation or computer science work yourself. Interferons and interferon pathways play key roles in the body's immune system. And one of the things that researchers have been starting to do is use these to help treat cancer. So anti-CTLA-4 is a very common immune therapy drug known as ipilimumab. And one of the most interesting things about it is that it's not successful in a lot of tumors. But in the tumors that it is successful in, it tends to be very successful and very efficient. Now, one of the hypotheses that's been posed as to why anti-CTLA-4 or ipilimumab isn't successful in all tumor types is genetic variation within the genomes of the tumor cells. And while that's been seen to account for some, there are still distinctions that we don't understand. So, my research is trying to explain the tumor lines that don't respond to anti-CTLA-4 treatment by looking at the variable mRNA expression or protein coding RNA expression of these interferon and interferon pathway genes. So I wanted to include a couple intracellular diagrams to demonstrate just how complex some of these interferon pathways are, because there are a lot of genes that are associated with this, and it's not simply one gene. And so that's one of the things that makes this kind of widespread research very important, and why the Cancer Genome Atlas gives researchers a really unique opportunity to look into expression of all of these different genes. So while I don't have time to explain the entire sequencing procedure for mRNA, suffice it to say that each of the individual reads, which you can see as those little itty-bitty lines, when aligned with the reference genome, gives a relative count of the number of mRNA strands in the sample. It's not an absolute count. There are X number of mRNA strands for this genes, but it gives a relative measure of the number of genes. So what I actually did this summer was I used proprietary software to interrogate these Cancer Genome Atlas datasets with focusing on interferon pathways. And I did that to see how survival curves change across different cancer subtypes as well as different cancer treatments. And I was looking for an RNA level biomarker that will help clinicians inform therapeutic choice to better treat a patient's tumor depending on the unique profile of that tumor. So on the left, you should be able to see a screenshot of what the kaplanmeier plotter tool looks like. And this is the tool that I used this summer. And so there are three things that are really important to understand about this. One, it allows you to develop what's called a cohort, or a group of patients. And you have full control over that cohort for a lot of different aspects of patient metadata. So things like where the tumor is located, whether or not there are other tumors involved, how the tumor was treated, what stage the tumor was caught at, a couple different identifying characteristics of individual patients like age. And so that's the first thing. And the second thing is that you're able to enter and interrogate dozens of genes at the same time. So I think I put in like 80 or so genes at one time and ran analyses on all of them concurrently, which was really convenient because it allowed me to interrogate a lot of cohorts without having to spend time looking at each individual gene in a given cohort. And then the third thing that's really important to understand about this tool is that it has a totally customizable output. It's primarily for Kaplan-Meier survival analysis, which you'll get to see some of those curves in the next plot. But you can also have access to things like B-swarm plots and a couple of other really important statistical significance things to ensure that the data that you're getting is the data that, one, you think you're getting, and two, is relevant and doesn't just appear to be statistically significant and actually is. So this is what the KM plotter actually outputs. So on the left, you can see the survival outcome and significance curves, where it demonstrates the probability of a patient's survival as time progresses, depending on whether they fit in the low or the high cohort. On the right, you can see the B-swarm plot. Now, the x-axis for these plots is fairly arbitrary, but each point represents an individual patient, and the y-axis represents the relative measurement of that gene's expression. So if you look in the top left corner, in the one that would correspond to IFNA1, you can see that any expression of that protein, IFNA1, would place you into the high-expressing cohort. Now, if you go back and look at the survival outcome and significance graph, you can see that being in the low-expressing cohort makes you about 60%-ish, I'm eyeballing it, so that might be a little bit off, more likely to survive past 150 months than somebody in the high-expressing cohort. Now, analyses like these can be really, really helpful when you do this based off of a specific treatment. So, if a specific treatment is better for low-expressing cohorts, this is what you're likely to see. So I looked at 6 different treatment pathways, and they were chosen either because they were immune therapies or because they have been demonstrated to cause immunogenic cell death and are at least partially dependent on immune response for drug efficacy. So there was a real variety of significance across different genes in these interferon pathways. One of the ones that I found to be really significant was interferon regulatory factor 4, which across all the cancer subtypes I investigated, high expression led to better survival for 4 out of the 6 treatments, which is, pretty serious. But, then you had some other ones like TRIM34, which were overwhelmingly insignificant, and the instances where it was significant, it wasn't very significant, and those instances of significance tended to be contradictory. So one of them, the high expressing cohort, would be more successful, and the other one, the low expressing cohort, would be more successful. And so this, and molecules like this, tend to bring up a lot of questions about things like cohort size, because some of the cohorts that I investigated were, no larger than like 20 to 40 people. So when you kind of get big tables of these data points, you can sort them in 2 different ways. One of them is tabular results by treatment, and so this mostly brings up scientific questions, and this brings up a lot of questions about mechanisms. So how does gemcitabine or cisplatin actually work within a tumor, rather than being truly clinically applicable? This brings up a lot of good questions about that you could propose further experiments based off of to demonstrate specific mechanisms for how these treatments actually work. So when you tabulate this data by cancer type as opposed to treatment type, you get data that paints a little bit of a different picture. So let's say that you had a lung cancer patient who demonstrated a high expression of JAK1 and JAK2 genes. You can see from the table below that high expression of those genes is associated with better survival under treatments like Promextred rather than cisplatin or carboplatin. And so physicians could use this to help inform therapeutic choice in terms of choosing which treatment is more likely to be successful depending on the individual profile and unique profile of these patients. And while it still brings up a lot of scientific questions, it's less mechanistically oriented and more oriented towards a clinical setting. So I mentioned this a little bit earlier, but one of the genes that really stuck out across all of my research tended to be interferon regulatory factor 4, which is a transcriptome factor. And it's found to be statistically significant and associated with better survival when expressed highly across pretty much all the cancer and treatment types that I investigated. So doing a little bit more digging into what this protein actually is and where it's found, it turns out that it's not actually found within the tumor itself, and it's usually found in immune cells that are found in the tumor microenvironment. And so even though it appears that these tumors have high expression of interferon regulatory factor 4, it's most likely the immune cells that are in the tumor, embedded in the tumor, attacking the tumor, that are actually expressing these proteins. And so one of the potential uses of this in a clinical setting was, rather than being an RNA level biomarker in terms of determining which treatment is best, is it could help determine in a much more efficient way whether or not the treatment plan that you're using, specifically when it's an immunogenic treatment like anti-CTLA4, is actually making a difference. And so if you don't find high expression of interferon regulatory factor 4, you might want to change your treatment type. And so this could potentially speed up a clinician's ability to say, well, maybe my treatment path that we've been working on isn't a good idea, instead of having to wait months and do tests to look at how much the tumor size changes over time and all these other different tests. They could potentially and much more efficiently determine in a matter of weeks with a very cheap protein assay whether or not their patient is responding to the treatments that they have them on. So there are a couple things that I really want to do in the future to build off of this research that I did this summer, and it's really investigate a lot of these correlative relationships that I established with either murine or ex vivo models. And murine are mice models, and ex vivo models is tumor cell lines like in glass. And so one of the things that we hope to do is use gene silencing or gene editing techniques in different cancer cell lines to attempt to confirm some of these associations and investigate variable responses to interferon pathway therapies in modified cell lines, as well as kind of working to develop a better understanding of interferon regulatory factor 4's role in the tumor microenvironment. So here are my citations for all the work that I used to either get some of these responses or help to develop my thoughts around these responses and shape how I looked at them and presented my data. A couple acknowledgments I want to make before I close this out. I wanted to thank Dr. Hargadin for helping me get this project, as well as building a very helpful framework for me to think about this project in a very intuitive sense. I wanted to thank Dr. Blaz-Giorfi for not only developing the KM plotter tool, but allowing us to use it for free. We really couldn't have done the research without him. And finally, I wanted to thank the Hampton-Sydney College Honors Council and the Office of Undergraduate Research for funding and supporting me through my research this summer. Thank you so much. I hope this was helpful and informative.